it's a great honor and privilege, obviously, to introduce Lennert Mucke from the Gladstone Institute and UCSF as our today's neurodegeneration seminar speaker. Although it's based in the Bay Area now, Lennert actually has a noteworthy history in San Diego, while he was based at the Scripps Research Institute, and then has had a very long and fruitful collaboration with our very own Eliezer Maslia before he left for NIH. In looking over Lennert's CV, I didn't realize how widely traveled he has been before settling down in San Francisco. Lennert was born in Germany, received his medical degree at the George August University in Göttingen in 1983. Following a brief one-year research fellowship in the Department of Neurobiology at the Max Planck Institute in Göttingen, he came to the US to train in clinical neurology, first as a resident in internal medicine at the Cleveland Clinic, and then as resident and chief resident in neurology at the Massachusetts General Hospital. He then moved cross-country for a postdoctoral fellowship in neurovirology with Mike Oatstone at the Scripps Research Institute. He remained at Scripps for a total of 8 years until 1996 before being recruited to the Gladstone Institute in San Francisco. 2 years later, he became the director of the Gladstone Institute of Neurological Diseases, where he has remained since, and a position he still holds. He is also the Joseph Martin Chair in Neurosciences in the Department of Neurology at UCSF. He has received numerous awards. I'll just mention several notable ones, including the Potemkin Prize for research in PIX-AD and related diseases from the American Academy of Neurology, and the MetLife Award in medical research from the Metropolitan Foundation, a Life Foundation. And in 2019, he was elected to the National Academy of Medicine. So truly a distinguished career that is notable for its sustained excellence. His initial research that received wide acclaim is the development of multiple lines of APP transgenic mice, most notable of which are the J9 and J20 lines, which he has characterized with both his usual rigor and creativity to study the pathophysiology of AD. But even more so, he has been remarkably generous in sharing this prized experimental mouse model with colleagues around the world, including to little old me. Most recently, he has turned his attention to brain network dysfunction in Alzheimer's disease, some of which we will hear today. He was the first to call attention to subclinical seizures in Alzheimer's disease patients, a finding that was initially quite controversial, but now widely accepted. Not only that, treatment trials with anti-seizure medicines are ongoing, and in particular, Keppra, or levetiracetam, has shown promise in improving cognitive dysfunction in AD research subjects in some trials. Finally, one measure of how good a lab is the mentoring history. In this regard, the Mucke lab has excelled in training exceptional researchers. I would just mention two individuals who trained as postdocs in his lab. The first is Tony Wiss-Correa, now at Stanford University, and I believe many in the audience is familiar with Tony's outstanding record in aging and AD research. The second is Li Gan, now the director of the Appel AD Research Institute at Cornell Weill Medical School. But most importantly, Li Gan is also the postdoc mentor of our very own Sue Chen in the Department of Neurosciences at UCSD, one of the newest recruits to our department. Clearly then a legacy of trainees who have gone on to outstanding careers of their own. So without much ado, thank you very much, Lennert, for agreeing to give this talk. We all look forward to your exciting talk. Take it away.
Well, thank you so much. Let me share my screen with you, and let me say also, how closely connected I feel to the UCSD department there because of my longstanding collaboration and still knowing Bob Terry and Leon, and I feel that I really got into this field of research through the environment that you guys have created there, which is so stimulating and wonderful. And so I really appreciate this and feel it's part of me in many ways. So let me share my screen and hopefully you will be able to see. Can you see my slide?
Yes, we can.
Okay, perfect. All right, so I'm going to talk about neural network dysfunction, very broadly defined, and just some disclosures which include some consulting and advisory board activities, membership in a startup company, some industry collaborations, some patents that are not held by me, but by my institutions. And whatever I'm mentioning in terms of drugs, these have not yet been approved by the FDA for their uses that I will discuss. To continue the disclosures, this particular lecture is focused on findings from my lab. And so I'm going to be very egocentric here and apologize to those whose important discoveries are not mentioned. If you're looking to hear about tau aggregation and propagation, you will be sorely disappointed. I won't be speaking about them. Be prepared for some surprising findings and unorthodox views. And my overall warning is that nobody truly understands neurodegenerative disorders yet. And I must say that the more I learn about them, the more perplexing and daunting I find them to be. Not just AD, but Huntington's, for example. I mean, I thought that ought to be a slam dunk, and it clearly isn't. So Alzheimer's disease, as you all know very well, has many players that range from A-beta assemblies to tau aggregating to astroglial and microglial responses. And your own Katerina Akasoglu, whom you nicely sent us as a faculty member, discovered that fibrin leaves the blood vessels when they are damaged in AD and triggers microglia via the CD11B receptor to become neurotoxic. There is ApoE, which in the E4 variety promotes amyloid deposition, but as Yadong Huang and Bob Maley showed, also becomes abnormally truncated and then impairs mitochondrial functions inside of neurons. And in many cases, as you know, over 50% of people have also alpha-synuclein accumulations in AD brains. And so I think anybody who thinks that there's one blockbuster drug that will take care of all of that, I think has another thing coming. So we need to be prepared for having to develop a diversified drug portfolio to address all this. Now, when you have such a scenario like this, it becomes, of course, important to figure out which of the various causes has the biggest impact on the disease, because that's precisely the one that you would want to target by therapeutic interventions. So how do you predict the relative impact of different factors into disease? And I think since it takes so very long to do this in humans, really takes phase 3 trials, experimental modeling becomes really important. And I would say that much of what we have done in the field and others suggests that 3 really important players are APP, tau, and ApoE4, not only from a genetic perspective for APP and ApoE4, but across various disease models. One should think that these are players to be taken seriously and that deserve a good effort in therapeutic targeting. But there are many other things, as I pointed out in the other slide. So my current working hypothesis, which is very simplified, of course, has in it APP. It ignores for now ApoE4, which is dreadful, but I just, can't talk about everything that I find is really important. ApoE4 is clearly a very important factor to further understand and target. But if we just focus on APP and tau, I think that non-fibrillar forms of these, of their metabolites, fragments, or the hollow proteins, very likely contribute to neuronal network dysfunction. I doubt that they are doing it via these pathological hallmarks, the amyloid plaques and the neurofibrillary tangles. I think they are on pathway, but kind of parallel, and therefore they may correlate with neural network dysfunction. But I'm not convinced, certainly, that amyloid plaques or even the tangles cause the neural network dysfunction that underlies cognitive impairments. Now, aberrant glial activities have emerged as a very interesting possibility from the GWAS studies. And I think there is a good chance that they more directly cross-connect with neuronal and network dysfunction. And I'll give you some examples of this in the talk. So looking at the APP molecule shown here, there are many different entry points surrounding it for therapeutic interventions. There is BACE, there is the presenilin complex, the gamma secretase complex, and there is work that Eddie has done together with Dale Bredesen and others that looks at the C-terminal domain, which also has some interesting caspase cleavage possibilities, and then there is aggregation and so on and so forth. So just focusing on this particular molecule, I would say that it has taught us humility as the solid foundation of all virtues. I think, you know, the more I learn about this molecule, the more astounding it becomes to me. So I'll take you a little bit into our model exploration of APP, which began a very long time ago, actually still in San Diego when we made some of the first APP transgenic models. Eddie mentioned the J20 model, which, you know, like many of these simulate some aspects of AD very faithfully, like the A beta that deposits the dystrophic neurites that you can see. And this is done by your very own Eliezer Maslia here in a human with AD compared with the mouse section. Now, the way I see APP mouse modeling is sort of like this. I believe that the conventional APP transgenic mice will overlap to some extent with human sporadic Alzheimer's disease, but there will be just transgene artifacts, overexpression artifacts, other things that will have nothing to do with the disease. But there is that sort of area of overlap from which we can learn something useful. There is possibly wider overlap with familial autosomal dominant AD because in many of those APP mice, we express these mutations. And many people are very preoccupied about overexpression, but I feel a little bit more encouraged by nature, who has done these overexpression experiments in humans through APP duplications and triplications and trisomy 21, well studied, you know, by Bill Mobley and others. So I think that here we have overexpression of APP resulting in Alzheimer's disease. So I think it's all right. And I keep asking the people that tout the knock-in models as so essentially different, what really is so different, what they find in the knock-ins from the overexpression models. And I see much more overlap actually than differences. So we set out to study this in earnest by comparing 3 lines. And this is work that Eric Johnson, when he was in the lab, spearheaded. And so we took 3 different lines of mice that are dubbed I5, J20, and knock-in here for short. The knock-in model is the one that Takaomi Saito and Takashi Saito developed in Japan at the Riken Institute. And this is a knock-in with a humanized A beta sequence, and it carries 3 different FAD mutations that are described here. Here is our mouse model with the Swedish and Indiana mutation, and then this one has no mutations. It's a wild type line we made. And of note, the APP isoforms that are expressed are all 3 major APP isoforms in our line. Whereas I would say that the knock-in mice probably express primarily APP695, the predominant isoform in mice. And it's often, I think, not appreciated that in humans there is much more APP751 than in mice. So you don't simulate that particular ratio of 751 to 695 in these knock-in models. Our models are driven with a heterologous promoter, and here is the mouse promoter, and they have all been previously described. But here we're really looking at them side by side. And so we're looking at the levels of hollow APP, we're looking at the amount of A beta that is produced, and we're also looking at the production of C99, which is created by this base cleavage, and C83, which is created by the alpha secretase cleavage. And so comparing these lines, this is just a Western blot where you can see that The wild type APP overexpressor makes quite a bit more than wild type APP. This is a human and mouse cross-reactive antibody here. The J20 mouse also overexpresses APP, and there is no overexpression, as one would predict, in the knock-in model, either in the hippocampus or the cortex. You can see that there is also quite a bit of C99 accumulation in J20, much more so than in the wild type counterpart. And in the cortex, interestingly, there is quite a bit of C99 accumulation also in the knock-in mice. And so this one shows you the comparisons. So this is a read of the full-length APP looking at both human and mouse here. And so the wild type mouse have the biggest level of APP overexpression than the J20 model, and the knock-ins don't overexpress APP. Here the C-terminal fragments in the hippocampus on the left and the cortex on the right. And you can see that here, as I showed you, the J20 makes more in the hippocampus than these other two lines. But here in the cortex, actually, the knock-in mice have as much C99 as the J20 model. So one clearly isn't just overexpressing a beta in these mice, but also actually some of the other fragments, in particular the C99 in the cortex, as shown here. Now, this is interesting if one looks at this study now of humans. So these are base-generated APP C99 fragments, and they are clearly elevated in autosomal dominant AD in brain tissue, but not really much, only a trend in this direction in sporadic AD. Now, what about the oligomeric assemblies? So we adapted an assay that Dennis Selko's lab developed, and I think is really very solid. We further validated this in this paper. If you look at the supplemental section, there is a lot of work on this in there. And so here you can just see the A beta production, and the levels are very high. So this is in young mice, but even at very early ages, there's already a lot of deposition. So I doubt that this is really soluble A beta. This is already partly from plaque deposition why this is so very high. And the oligomer ratio, here's the A beta ratio on this side, which you can see here. And then these are the oligomer levels. And you can see that in the hippocampus, the J20 mouse has more oligomers than the knock-in, but actually in the cortex, there is more oligomeric A beta in the knock-in than in the J20 mouse. So, you know, you're looking at these and they're obviously they're making more A beta and they're making more A beta oligomers, but the distribution is clearly a little different. And so is also the timing of it all. When one looks at the behavior of these mice, they all develop behavioral deficits, but the knock-ins are delayed. This is an active place avoidance test where the mice have to use extra maze cues to find out which particular part of a circular platform is aversive with electroshocks, and they learn how to avoid this. There are less and fewer entries into the aversive zone here, wild type controls. But you can see that even the overexpression of APP in non-mutated form impairs the mice. And so the J20 is impaired, and there's a little less impairment here, but still, if you quantitate it, all the different lines are impaired. And here is just a different readout, the maximal avoidance time, where they're all impaired. There are some differences, interestingly, when you look at wild type controls from the different lines. So it's always really important, I think, to use wild type controls from the same line. I think Takaomi often doesn't do that and just uses homozygous breedings within a line and then a different wild type control. That can be a little bit misleading, but so in any case, they're behaviorally impaired if one waits long enough. So here, 8 to 11 months. And they also all have hypersynchronous network activity as shown here in the form of epileptiform spikes and spike and wave discharges, which are shown here. And you can see the quantitations. So the J20 model clearly has more of these just singular interictal spikes than the knock-in mice, but the knock-in mice clearly have a lot more of these sharp wave discharges that you can see here in illustration. Now, is this relevant to the human condition? And Eddie already alluded to the study that we did, which was led by Keith Vossel and our colleagues at UCSF in the Memory and Aging Center and the Epilepsy Center together. So we really wanted to know what the percentage of people with early Alzheimer's disease, sort of in their 60s, is in terms of epileptiform activity. So we took people who had no known seizure history and age-matched controls, admitted them overnight for long-term monitoring by EEG, and then in the next morning, they had a 1-hour magnetoencephalogram with simultaneous EEG recordings and some clinical assessments. And so what we found is that quite a few of the patients showed very subtle, but based on review by 2 neurophysiologists and 2 epileptologists, by consensus, they picked out things like this sharp discharge here in the central area in one patient, and here maybe a more prominent epileptiform event that cuts across multiple channels in the magnetoencephalogram of this person that was not seen at all in the surface EEG that was recorded simultaneously. And in terms of location, as one might predict from where the pathology starts, much of this was clustered around the frontal and temporal region. Now, looking at the incidence, over 40% of the people we looked at, which are relatively young in the early 60s, had epileptiform events. And we saw it also in 2 of our control patients. So I don't know whether that's just what you see in a normal aged population, or if they will sooner or later move over into this pie. But in any case, the preponderance of epileptiform, subclinical epileptiform activity was clearly much greater here in the AD cases. And I should say that this has been nicely replicated by others. So this is from Alice Lamb and the group at Mass General Hospital, where they also find very similar associations of epileptiform events with Alzheimer's disease. Now, they did a really cool study in 2 patients where they were bold enough to place foramen ovale electrodes, which sit then right next to the temporal lobe where the action is in patients, and they compared them with either surface EEG SE or the foramen ovale electrodes, either in awake state or when patients were asleep. And in both of these patients, you can see that the foramen ovale electrode picks up many, many more epileptiform spikes than the surface EEG. And that is easier at night than during the day, which is clearly also our experience from our clinical study. And here's another study from Budapest that came out that I think is very interesting, where they compared the sensitivity of surface EEG recordings in picking up epileptic activity in patients with probable AD. And they basically demonstrate then at night, a 1-hour recording at night is as sensitive as an 8-hour recording during daytime. So most of this activity can be detected when patients are asleep. That is, I think, important to note if one just wants to look for them by surface EEG. Keith, when he looked at, Keith Vossel, when he looked at the patients longitudinally over time, also found that the people in whom we could detect epileptiform activity had a much faster decline in mini-mental state exam score and also in a global executive function score than those without. And that raises the question whether those have a more severe type of Alzheimer's disease, or if the epileptiform activity actually contributes in some way to the cognitive decline. Now, one should notice this was an earlier study, more retrospective and observational we did, where we looked at people who actually were seen at UCSF for both epilepsy and Alzheimer's disease. And it turned out that really the majority of these events were non-convulsive and could easily be missed if one isn't in tune to looking for them, consisting of jamais vu or déjà vu experiences, psychic sensory phenomena, staring spells, speech behavioral arrest, amnestic spells, or confusional episodes, altered consciousness. And what was also remarkable, whereas many people sort of think of epileptic events in AD as a late stage burnout phenomenon, the onset of these epileptic manifestations clustered, I would say, around the onset of cognitive decline, but then could occur really at any stage afterwards. So I think that all in all, I feel that there is a great need to put more emphasis on functional network assessment in biomarker discovery. A while back, I attended an interesting workshop at the National Institute of Medicine where they had brought together researchers who looked for biomarkers in Alzheimer's and in schizophrenia. And it was like these two groups were, came from completely different camps. One obsessed with morphological changes, the other with all functional things like, certain waves in EEG. And it was really interesting. I think a merger would be of benefit here clearly, certainly for AD, where there are lots of things where one could probably make interesting discoveries by looking at more functional readouts. Of course, all the biomarker discovery is fascinating, including in blood and CSF and all of that. So now what about therapeutic interventions? And as you know, this has been really a very, I would say, humbling experience for the field, where everybody thought after the identification of gamma secretase and base that this was sort of, nailed, and now we just need to develop the drugs and we're done. And both of the gamma secretase and base inhibitors have failed pretty miserably in clinical trials. But we wanted to use base inhibition as a tool compound to really assess to what extent the alterations we find in APP mice are dependent on the production of A-beta and also dependent on the production of C99. And so we obtained a base inhibitor from Novartis, and we put it in the chow of the mice and treated them for, several months or just a few weeks. And what we found was that it was incredibly effective. This is the duration of treatment down here, either a placebo chow or the base inhibitor chow. And then we're looking at A-beta 1-2X. Can see very profoundly reduced in the hippocampus. Here is A-beta 1-42, also practically wiped out. Also A-beta oligomers, something that I wasn't quite sure whether that would necessarily be the case. But clearly, if you shut off the source of the A-beta oligomer formation, you also lose the A-beta oligomers after the treatment. So that was also very encouraging. So we get rid of the A-beta forms and we get rid of the A-beta oligomers. We also, as you would predict with the base inhibitor, drastically reduce the production of C99 as shown here, but you do get an increase in C83 as a result of that. So, it's funny with APP that you can't do anything to it that's just one thing. You always get a multitude of other things change at the same time, potentially confounding interpretations. And so here you're not only reducing one thing, but you're also increasing another. And so goes the game that surrounds APP and, by the way, many other AD-related factors. Now, we were quite surprised to find that the very effective reduction of A-beta oligomers and C99 really did nothing to the what you might call clinical manifestations or preclinical manifestations in the mice. The mice have a premature mortality that's almost certainly related to their epileptic activity, and that was entirely unchanged even when we initiated the BACE inhibitor treatment at 1 month of age. There's a lot of discussion about how early might you have to go after A beta or APP, and so here we went after it pretty early. And the same with the epileptiform activity. And the same with a variety of behavioral readouts. So here is the Morris water maze training phase in impairment in task acquisition as shown here and quantitated further here. Absolutely no movement after the BACE inhibitor treatment on this. And here also in the probe trial, we see nice preference for the target quadrant in the controls. Now, different from the human trials in which BACE inhibitors have failed as well, we don't really see an impairment of the non-transgenic mice by the BACE inhibitor. Maybe a tiny bit, but it's not really worth writing home about. But clearly there is the APP-related impairment, and the BACE inhibitor does nothing here to improve it. And this is what I showed you before, the active place avoidance, again, impairment in the J20 mouse, and nothing happening there. So, how does one interpret this? Either there is something very peculiar in this line that, causes these kinds of deficits independent of APP. We consider this pretty unlikely, but we haven't tested other lines with that same BACE inhibitor because I can handle only so many negative results at any point in time. And so I might muster up the courage to look at another line with this, but clearly here BACE inhibition was ineffective, which to me says that at least in this one model, the deficits are probably not dependent on A beta or C99 production. Now, what else could they be? I think there are some interesting signaling possibilities of APP when it's mutated that Eddie and Dale have worked on. And so I think that that's worth a consideration, but also the oligomers don't seem to be driving the neuronal dysfunction and cognitive impairment in this line. So I want to understand APP. There is no question that it is important in Alzheimer's disease. The genetics are very clear, and so are many other lines of evidence. But I'm beginning to doubt that it works the way we're thinking it works. And the clinical trials certainly are in line with that, I would say. So let me move over to this other player, one of my other favorite molecules, equally enigmatic in many ways. As you know, tau is a very complex gene that undergoes alternative splicing, and in the human brain gives rise to 6 major isoforms that are shown here. But the complexity doesn't end there. It has many post-translational modifications. And I would argue that we don't understand the physiological function of tau. Initially, it was viewed as a stabilizer of microtubules. I seriously doubt that that's what it does. In fact, I can't imagine that's what it does, because you can knock it down in an adult brain that has lived with tau all its life, and not much happens with microtubules. And there are many other lines of evidence. I think it's more likely to me that it participates somehow in the formation of signaling microdomains, perhaps, and is involved in signaling cascades somehow. But if anybody tells you they know what tau does in the brain of an adult or aging animal, I wouldn't believe them. There are tons of post-translational modifications, even of physiological tau, over 60 of them. And I don't think we really understand what their purpose is. Some of them are modified by temperature of the body, by salt intake, and so on and so forth. Initially, I think tau was viewed primarily as an intracellular molecule, and that certainly is there, mostly in the axon, but it also has an extracellular life, probably mostly in truncated form. It is released from cells. You can find it, as you know, in the CSF. It's not only made by neurons, but it is made also by glial cells, particularly by oligodendrocytes. And I don't think we understand what the role of tau is in these other cells either, but it's interesting. It's also, to me at least, entirely unclear what the most pathogenic form of tau is. There are many different species that have been identified. Is it an aggregate? Is it a conformer of a monomer? What is it? There is a plethora of downstream molecular pathomechanisms that have been described, and this is a recent review we published in which we're really going at length into many of those things. And then there is almost an obsession in the field about spreading of tau from one neuron to the other, and the propagation of aggregated tau. And for the life of me, I can't find any evidence in the literature that the tau that spreads and is taken up by another neuron impairs that neuron in any shape or form, either the function of the neuron or its survival. I can't find that evidence in the literature. Yet, as a phenomenon, it's interesting, and many people are interested in studying it. So here are three key ways of how tau could lead to neuronal dysfunction and degeneration. In the early days, people speculated that perhaps the aggregation of tau in neurons would sequester physiological tau away from microtubules, destabilizing them. I don't think this is a likely scenario because, again, you can really reduce it in mice and flies and in non-human primates, and now in the clinic in Alzheimer patients, without seeing any detrimental effects out of this. But this is certainly what people are most focused on, which is that tau somehow, by aggregation or in some other way, assumes an adverse gain of function that contributes to neuronal degeneration and dysfunction. And I think that is very likely. And you know, how exactly that goes is not so clear. In fact, people have described many, many different pathomechanisms that all make sense, for all of which there is some evidence. And it gets complicated because, you know, there are all these different assembly states, and some of them may be toxic and others innocent bystanders, and some of them perhaps even protective. So here I want to actually focus on this third possibility that I've become particularly interested in, which is an enabling role of tau that might have to do with its physiological function, but in disease contexts, it might become contributory. And so let me just highlight this in a very simple diagram, for which I apologize. So, you know, the idea here is that there is a pathogenic trigger, something other than tau, that drives pathways that are connected to tau, and that this pathway drives neuronal dysfunction and degeneration. But because of this link here, you can indirectly manipulate this. And so let me give you one example of this that we published a while back, and this is work that Eric Roberson did. This shows you the epileptiform activity in the J20 model on a tau wild type background. So there is no human tau overexpressed. This is just endogenous wild type tau. When you cut the tau in half in a heterozygous knockout background, you greatly diminish this aberrant hyperexcitability. And when you completely remove tau, there is none of it left. So APP here can induce this kind of neural network activity only in the presence of tau, and it needs at least 50% of it, probably a bit more, for this to occur. So here's APP, and here is tau, an enabler in this sense. Now, we thought initially that maybe APP would be changing the post-translational modification of tau or something else. So we collaborated with Al Burlingame at UCSF and did a pretty extensive mass spectrometry study of mouse tau. Just wild type mice have at least 63 different post-translational modifications, from methylation to acetylation to ubiquitination or gluconylation, you name it, and of course all the phosphosides. And we didn't find any differences in the APP-J20 models. So the idea that you would sort of have APP and it would take tau and turn it into a pathogenic mediator, we can't find a pathological tau species in APP mice. Now, there could still be something else, but we haven't found it yet. So we're kind of sticking with the enablement idea. So I'll give you some examples of what tau reduction can really accomplish in the setting of APP mice by looking at this paper that Melanie Das led, and we recently published in iScience that focuses on APP mice that are over 20 months of age. And you can see here with an anti-A-beta immunostain that the cortex and the hippocampus is really laden with amyloid deposits. And what we show is that tau reduction and anti-epileptic drug treatment suppresses the epileptiform activity, and that this actually alters the profile of innate immune cells in the brain, and that some of these alterations may be maladaptive, but some, for instance, increases in TREM2 expression are probably adaptive changes, where microglia may be using TREM2 to suppress the epileptiform activity. So to illustrate this for you, so we're looking here at either wild type tau mice or tau knockout mice that either do not express APP or express APP. So 4 different genotypes, and you can see by EEG recording, and these are pretty ancient mice, that the APP mice on the wild type background, they've got epileptiform activity, but on the tau knockout background, as I showed you before, they have hardly any of it. Now, this is now a neuroinflammation panel nanostring profiling of the hippocampus. And if you just compare APP mice on the tau wild type background with non-APP mice on the tau wild type background, you can see that some genes like HOMR1, for example, and other synaptic proteins are, transcripts, I should say, are reduced. And a lot of inflammation-related transcripts, some of which you may know well, like complement factors and TREM2 and DAP12, they are increased in the APP mice. Now, when you reduce tau or ablate tau in the APP mice, you shift this back over to the left, where all these inflammation-related genes are now brought back to where they once were. So again, APP can only induce these alterations of microglia macrophage products in the presence of tau, but not in the absence of it. Now, our working hypothesis is that the mechanism isn't some kind of molecular tau interaction with microglia or so, but that it's really driven by the network abnormality. And so we tested this hypothesis by treating the mice starting at 22 to 26 months of age with levetiracetam for 4 months. And we did 2 EEG recordings, and in this interval, they were tested behaviorally. And so just like the tau, but maybe not as effectively, the levetiracetam treatment reduces the epileptiform activity in the APP mice, and it also improves their deficits in the active place avoidance. We've previously shown benefits of levetiracetam treatment in much younger mice, and as Eddie alluded to, Michaela Gallagher is doing a fairly large phase 2/3 trial of levetiracetam, and Keith Vossel just published a very small trial of levetiracetam in people with Alzheimer's disease. Now, again here, just comparing APP mice with wild-type controls on the placebo, we see again that many of these microglial inflammation-related gene products are increased in expression in the mice. And when we treat them with levetiracetam, we shift things over to the left, just like with the tau reduction. And in fact, when we look at this, so this is the 27 genes were modulated by APP in one direction and by levetiracetam treatment in the opposite direction. And this is a string network analysis of the corresponding proteins. You can see that there are probably many things that you might be interested in the context of inflammation and Alzheimer's disease. As you know, TREM2 and TYROBP are fairly strong risk genes with a strong impact on AD, even though the variants that have been implicated are exceedingly rare. And there is good evidence that complement factors may be involved in abnormal synaptic pruning in the disease. Now, remarkably, the levetiracetam treatment had no impact whatsoever on the amyloid plaque load or on plaque-associated microgliosis. So just looking at IBA1 staining, we find as many microglia, and they look as activated as in the placebo-treated group. So we don't think that the difference is really in changing the response of the microglia in terms of recognizing the amyloid, but their molecular profiles clearly change. Mostly perhaps of the plaque-associated microglia, we did this by RNA scope analysis, but also perhaps in microglia that are away from the plaques. So when one then looks at this interesting overlap, so these are APP-dependent gene expression changes that were prevented by tau ablation. And these are APP-dependent gene expression changes that were reversed by levetiracetam treatment. And so those two treatments modulate 15 gene products that are shown here. And by string analysis, again, we come down to these kind of inflammation-related markers. And some examples we've then confirmed in independent cohorts of mice by RT-PCR analysis of hippocampus, of cortex. So they're not just seen in the hippocampus as we did with a string analysis, but also in the cortex. Here you can see the effect of tau ablation on C1qa and CLEC7A. And here you can see the effect of levetiracetam treatment on those two gene products. Here, TREM2 and TYROBP, same deal, tau reduction, levetiracetam treatment, very similar effects. In different cohorts of mice, we see beautiful correlations between the level of TYROBP expression and the epileptiform spikes. Just very nice. So let me return to the question whether these inflammation-related changes that we see in the brain of Alzheimer's disease and APP models are adaptive or maladaptive. Are they pathogenic or helpful in some way? And Tony Viscore, whom you mentioned, and we published a review back, I think, in 2000 or 2002. It was entitled The Double-Edged Sword of Inflammation in Alzheimer's Disease. And we really wanted to highlight that some of the changes that one sees are probably beneficial, which makes it really difficult to develop therapeutic interventions. They have to be very decisive so as not to backfire and go off in the wrong direction. So we wanted to understand a little bit what's going on with TREM2 because there is great interest in this particular myeloid cell gene product, and that's a fascinating one. So what Melanie did is she recorded from mice that are either wild type for TREM2 or heterozygous knockouts. And there's a lot of literature suggesting that the TREM2 variants that increase risk for Alzheimer's disease are hypofunction variants. So we are simulating here a TREM2 hypofunction by homologous knockout of one TREM2 allele. There is no APP overexpressed or anything going on. The only manipulation here genetically is the reduction of TREM2 levels and function. And that in itself is enough to increase epileptic activity in these mice at baseline. And when we challenge the mice with kainate, and then look at the amount of induction, we see an exaggerated induction, and this is a subthreshold dose of kainate. So really, you don't see much of this. I'm sorry, yeah, it's a subthreshold dose that affects the wild type mice less than you might perhaps expect with a full dose of kainate, bringing out this differential susceptibility. To illustrate this further, if we're comparing the TREM2 mice here, I lost one of the indicators, it's the gray, that's the TREM2 wild types, and then the TREM2 heterozygous knockouts are green. You can see after the kainate injection, a much greater response, and then it also kind of takes longer for that to subside over the subsequent days. So basically, we think here that the TREM2 hypofunction could promote network dysfunction because TREM2 may be used by microglia cells to counteract this kind of aberrant network activity, suggesting that in Alzheimer's disease, there may be a vicious cycle that can be either triggered by network dysfunction, for example, through a microinfarct with pleds surrounding it or some such thing, or by immune dysfunction where people who have low functioning TREM2 on their microglia are not able to suppress this effect and more likely to trigger this kind of vicious cycle that could contribute to synaptic dysfunction and loss. And you can block this perhaps with anti-epileptics and also with tau reducers. So let me say a few words about the mechanism of action of tau, which I must admit we haven't clinched yet, but I'm dying to find out how tau reduction blocks hyperexcitability of diverse causes. If it's APP, or it's a sodium channel mutation or a potassium channel mutation, you lower tau, you make the brain more resistant to network hypersynchrony. And so Chei-Wei Cheng in my lab did a cell culture and animal study focusing just on wild type and tau knockout mice. And what he found is that tau reduction affects excitatory and inhibitory cells differentially. It reduces overall the excitation to inhibition ratio and counteracts network hyperexcitability. And so I'll give you just an overview of this, and this was recently published in Cell Reports. So looking at a cell culture and doing microelectrode array recordings from 16 different channels, you can look at all these sort of action potentials that happen across your culture, and you can calculate a global synchrony index where exciting the culture with potassium chloride increases the synchrony. It creates hypersynchrony. It's sort of, a cell culture dish equivalent of what epilepsy might look like in a more complex network. And what we see in the tau knockout mice is that they have less of this hypersynchrony upon potassium chloride stimulation than the wild type cells. All the differences that I will show you are quite subtle, but they, I think, amount in the end to the shift in EI ratio. When we look at pyramidal cells, this is now in cortical slices, we can see that the tau deficient excitatory cells fire fewer spontaneous action potentials. And we also see when we record from fast spiking interneurons that they receive less excitatory input from excitatory cells. So that clearly should diminish the E in the EI. When we look at interneurons, we can see that there is increased intrinsic excitability of interneurons that are tau deficient, where a certain amount of current injection leads to more spike generation in the interneurons. And when one looks at the pyramidal cells and the input that they receive from the interneurons, you can see that they receive a greater amount of inhibitory input, which should increase the I in the EI. So the overall outcome of this is a shift in the EI ratio. And so here, as I showed you, if you've got something driving up the EI ratio like APP and you stop tau, you block the pro-excitatory aberrant effect. Now, notably, this works also in models of autism, for example, which are also associated with an increase in the I ratio. And Chow Tai, when he was in my lab, showed that in Dravet syndrome mice, which have epilepsy and develop signs of autism, like excessive stereotypic movements, the reduction of tau in half eliminates this autism-related phenotype. And again, our argument would be that the effect here is a shift in the EI ratio. Now, how do we treat tau? And this, I'm almost done. There are many different things that are being pursued, how to get rid of tau. And my feeling is that since it's so diverse from here on out, I want to go as upstream as I possibly can go. And I like the idea of going after the tau production much more than taking an antibody and going specifically after an extruded tau species, mostly in the extracellular environment, because it's but one of many tau species that may create all kinds of problems in the cell and, you know, in the whole network. So I think that for many diseases, the notion that has been pioneered by Don Cleveland and others at UCSD and the folks at Ionis Pharmaceuticals is very wise. Go at the upstream triggers so that you can try to prevent having to understand this very complex network that happens downstream of just about anything in the nervous system. And so if you can block the upstream trigger, that's the way to go. And I think that's all the more important because I think it really is uncertain whether the most pathogenic form of tau is a misfolded monomer, a fragment, a multimer, fibrillar or non-fibrillar. And anybody who claims they know this, don't trust them. Whether it acts intracellularly or outside of the cell, which of tau's many post-translational modifications, conformational states and isoforms are most critical, and whether tau spreading actually impairs critical brain functions or neuronal survival. So I think the most pragmatic approach here is to develop strategies to reduce overall tau levels. And I think that way you should be able to get both the sort of adverse gain of function mechanism as well as the enablement mechanism that I outlined to you before. I think both of those are of interest, and overall tau reduction will get them. Now, Tim Miller has pioneered this with Ionis very beautifully, showing that an ASO targeting tau is well tolerated and improves the survival and reduces behavioral deficits in a mouse model of frontotemporal lobar degeneration, the PS19 mouse model, and this has moved into a phase 1 trial, which has been well on its way without any safety signals. And hopefully sooner or later we will hear if there was any benefit from this. However, I think just conceptually thinking about the ASO approach and small molecules, I think aging populations, intrathecal injections of ASOs pose some challenges requiring regular visits. It's pretty expensive. There is much more experience with small molecules. There are very few successes, in fact, recent failures in SOD1 and in Huntington's disease. And I think small molecules, in my view, are going to be a better long-term survivor. So many examples of success in aging-related conditions of small molecules, not an AD yet, of course, but so we're very eager to develop small molecule tau reducers and are actively pursuing this together with Takeda. So let me end by revisiting my simplified working scheme. So here it is, reducing tau reduces both aberrant glial activities and the neuronal network dysfunction. This can also be done by levetiracetam, and I bet there could be glial targeting strategies that may achieve the same thing. In the end, I believe we will treat Alzheimer's disease like we treat other complex disorders such as atherosclerosis through a combination of different drugs. So I'm very pleased with the increasing diversification of the drug development portfolio. I think it's all worth pursuing and testing rigorously. There are big challenges to clinical trials. The recent CTAD meeting, highlighted that beautifully. We can talk about it if there is time, but let me end here. My current group and some of the cores that support us are shown here. I mentioned the folks that have driven most of this work. We have outstanding collaborators at UCSF and Gladstone, and thank you for the reagents and mouse models and for the financial support. And I'll end here, and I don't know if there's any time for questions. Take care, everybody, and thank you so much for the kind invitation.